Welcome to The Explainer. Today, we're going to unspool the true restorative history behind what is arguably one of the greatest scientific discoveries of the 20th century. We're talking about the literal instruction manual for life itself. But more importantly, we're going to look at the brilliant mind that was left out of the textbooks for far too long.
Let me just start off by asking you a very fundamental question. Who discovered DNA? If you think back to your high school biology class, you probably have an answer ready to go. You picture a specific scientific model, and you likely picture a very specific pair of scientists. But is that really the whole story?
Okay, let's dive into this. When people talk about the discovery of DNA, you almost exclusively hear two famous names, James Watson and Francis Crick. They're internationally famous for discovering the double helix structure of DNA. But there is a missing name, a massive part of the story that went totally untold for decades. Because without the unsung hero, Rosalind Franklin, Watson and Crick might never have figured it out at all. Her contribution was absolutely crucial, yet it was massively overlooked.
To understand exactly how this happened, let's step back in time. Section 1. The Great DNA Mystery. So, picture the scientific landscape of the 1950s. Scientists actually already knew that DNA existed, that it was the literal instruction manual for life. But its physical shape remained completely unknown. It's kind of like knowing a book contains all the secrets of the universe, but the book is locked shut and no one knows what the key even looks like. This brought about the ultimate scientific puzzle. Researchers globally were desperately trying to answer three massive questions. First, how is DNA structured? Second, how does it actually store this complex biological information? And third, how can it copy itself to pass that information on? Solving the structural mystery became one of the most intense and consequential scientific races in human history. Literally every top lab wanted to be the first to crack the code. Which brings us to our next chapter, Section 2. Enter Rosalind Franklin.
While a lot of scientists were furiously building theoretical models, Franklin approached this puzzle like a meticulous scientific detective. She was a brilliantly talented chemist working at King's College London, and she had a very specific, highly technical skill set that would ultimately blow the case wide open. Her secret weapon? X-ray crystallography. Now, this is a method used to study incredibly tiny molecular structures by analyzing exactly how X-rays bounce off of them. Without getting bogged down in the complex physics of it, just know that this technique allowed her to study the invisible. And Rosalind Franklin wasn't just good at this, she was exceptionally good at it. She possessed the extraordinary patience and precision required to get clear images of molecular structures that literally no one else could capture.
Her unparalleled skill sets the stage for our third section. Section 3. The Power of Photo 51.
All of her meticulous preparation, all those hours adjusting equipment and analyzing bounced X-rays in her lab were building up to one specific moment, the ultimate smoking gun in biology, Photo 51. Now, what's really interesting about this slide is that this image, captured by Franklin in 1952, remains one of the most important scientific images ever taken in human history. It wasn't just a photograph, it was a total revelation.
Let's break down exactly what this photograph achieved. Photo 51 revealed a distinct, undeniable X-shaped pattern, and to a trained crystallographer like Franklin, that specific X pattern provided groundbreaking proof of a spiral shape. It was the biggest, most undeniable clue to DNA structure. The mystery of what the instruction manual of life looked like was finally coming into focus. It was a spiral.
But this incredible triumph leads us directly into Section 4, the double helix controversy.
Laying out the objective facts here really underscores the historical controversy. While Franklin was doing this precise, groundbreaking work in London in 1952, James Watson and Francis Crick were over at Cambridge basically hitting a wall. But then a pivotal event occurred. Watson was shown Franklin's Photo 51, and this was without her direct permission. That image immediately helped Watson and Crick realize DNA is a double helix, and using her data alongside other research, they built their famous DNA model just a year later in 1953. Tragically, in 1958, Rosalind Franklin passed away from cancer. And then, in 1962, Watson and Crick were awarded the Nobel Prize, becoming world famous for the discovery.
I want us to just pause on this number for a moment. 37. Rosalind Franklin was only 37 years old when she died. It is a heartbreakingly premature end to a brilliant life. Because she passed away before the Nobel Prize was awarded in 1962, she was completely excluded. You see, Nobel Prizes are not awarded posthumously. However, historians widely acknowledge that her indispensable contribution was never fully recognized during her lifetime by the very people who relied on it. This tragically fueled her exclusion from the history books for decades to come.
But her story definitely doesn't end in the 1950s.
Section 5. A lasting scientific legacy.
So the crucial point is that this isn't just a history lesson about who got a prize and who didn't. Without Rosalind Franklin's research, the double helix might not have been discovered so quickly, which means modern genetics could have developed much, much later. Her work directly helped open the door to DNA testing, genetic engineering, modern medicine, the entire field of biotechnology, and even crime forensics. Literally everything physically shaping our modern world today traces its roots back to that foundational understanding of the double helix, and that understanding relied entirely on her data.
More than 70 years later, history is finally beginning to correct itself. As one modern retrospective noted, she's now remembered not just as the woman behind DNA, but as one of the most important scientists in modern biology. She captured the critical evidence, she provided the key clue, and today, she is finally receiving the massive recognition she has always deserved.
But Rosalind Franklin's story also reveals something much bigger about the way we record scientific achievements. It forces us to ask this final question: who else has history forgotten? How many other brilliant women in science were overlooked, ignored, or just quietly left out of the history books? As we continue to learn about the building blocks of life, let's make sure we never forget the people who actually did the building. Thanks for joining me for this explainer, and keep questioning what you think you know.